Welcome to our lecture on embryonic pluripotent stem cells. Imagine you have a magical block of clay that has the potential to be sculpted into absolutely anything, like a coffee mug, a detailed statue, or a delicate vase. In the human body, embryonic stem cells act exactly like this magical, unshaped clay. Today, we're going to explore what these cells are, how they work, and why they hold the ultimate key to the future of regenerative medicine. Let us dive in and uncover the master blueprint of life. Now that we have introduced the idea of a magical clay, let us look at what this means biologically. When a human life first begins, it is just a tiny ball of cells. At this very early stage, these cells have not yet decided what they want to be when they grow up. They hold the incredible power to transform into a heart muscle cell that beats, a neuron that thinks, or a skin cell that protects. We call this remarkable ability to become almost any cell type pluripotency, which literally translates to having many powers. Understanding these cells is like finding the master blueprint of the human body, allowing us to see exactly how a single microscopic entity builds an entire complex organism. Building on this idea of unspecialized cells, let us pinpoint exactly where they come from. In formal biological terms, embryonic stem cells are undifferentiated cells, derived from the inner cell mass of a pre-implantation embryo. This occurs specifically at what we call the blastocyst stage. To break that down, the term undifferentiated means they do not have a specialized function yet. Unlike a mature liver cell that processes toxins, or a red blood cell that carries oxygen, these early cells are a blank slate, waiting for the right biological signals to tell them what to become. Understanding their origin brings us to a crucial distinction in stem cell biology. The term pluripotent is very specific. It means these cells can differentiate into any of the three primary germ layers: the ectoderm, the mesoderm, and the endoderm. These layers eventually form every organ in your body. However, there is a limit. Pluripotent cells cannot form extra embryonic tissues, such as the placenta. This specific limitation is what distinguishes them from totipotent cells. Totipotent cells are found even earlier in development and have the power to form absolutely everything, including the placenta required to support a pregnancy. Now that we know what pluripotency is, we must ask how these cells maintain this special state. These cells are characterized by their unique ability to divide indefinitely in a laboratory setting while maintaining their unspecialized, blank slate status. This incredible property is known as self-renewal. But how do they do this? They are governed by a core network of specific transcription factors, primarily named OCT4, SOX2, and NANOG. Think of these factors as the cellular managers. They constantly work together to suppress differentiation, ensuring the cell does not accidentally turn into a specialized tissue before it is supposed to. With a solid grasp of their biology, let us look at why this matters to modern medicine. The discovery and isolation of embryonic pluripotent stem cells have completely revolutionized biological research. Because these cells can become any tissue in the body, they hold the ultimate key to regenerative medicine. They offer a potential way to replace damaged organs or tissues that simply cannot heal themselves. For example, if a patient suffers a spinal cord injury or develops Parkinson's disease, doctors could theoretically grow new, healthy neurons from stem cells to replace the dead ones. Furthermore, they are incredibly valuable for drug testing. Scientists can grow human heart cells in a dish and test new medications on them safely, without risking harm to a living human patient. To truly cement these biological concepts, let us look at this process visually. On the left side of the diagram, you can see the blastocyst. Notice the outer ring called the trophoblast and the crucial cluster inside labeled the inner cell mass. This inner mass is where we harvest our embryonic stem cells. Following the arrow to the center, we see these cells being cultured in a laboratory petri dish, maintaining their unspecialized state. From there, the diagram branches out into three distinct paths, representing the three primary germ layers. The top path shows differentiation into the ectoderm, forming complex neurons. The middle path leads to the mesoderm, creating strong muscle fibers. Finally, the bottom path shows the endoderm, forming internal organs like the pancreas. This single image beautifully captures the essence of pluripotency. Having seen the biological potential of these cells, we must now understand how to grow them in large quantities. When scientists grow embryonic stem cells in a laboratory, they need to know exactly how fast these cells are multiplying to plan their experiments. Because stem cells undergo self-renewal, one cell divides into two, two into four, and so on. This follows a classic pattern of exponential growth. We can predict the future population of our stem cell culture using a standard mathematical model. This is not just an academic exercise. This math helps researchers ensure they have exactly enough cells to differentiate into the required tissues for critical medical treatments. This brings us to the mathematical equation that models this exponential growth. The formula comes from the simple fact that every cell divides into two, which is why we use a base of 2. Let us break down every symbol. N of T represents the final number of stem cells present at a given time. N0 stands for the initial number of stem cells that we started with in the culture dish. The variable T represents the total elapsed time that the cells have been growing. Finally, the variable D represents the doubling time, which is the specific amount of time it takes for the cell population to exactly double in size. If the total time T increases, the final number of cells will grow exponentially. Conversely, if the doubling time D increases, it means the cells are dividing more slowly, resulting in a smaller final population. Now that we have our formula, let us see this with a concrete example. Imagine a researcher starts with an initial population of 1,000 embryonic stem cells. These cells have a doubling time of 24 hours. We want to know how many cells will be in the dish after 72 hours. First, we calculate the exponent by dividing the total time by the doubling time. 72 divided by 24 gives us exactly 3 complete generations of division. Next, we substitute this back into our equation, giving us 1,000 times 2 to the power of 3. 2 to the power of 3 is 8, which accounts for the compounding growth. Finally, multiplying 1,000 by 8 gives us our answer. After 3 days, the researcher will have successfully expanded their batch to 8,000 embryonic stem cells, ready for tissue engineering. Once we have successfully grown a massive population of cells, we face a new challenge. The next step is often to force them to differentiate into a specific cell type, such as motor neurons, using chemical signals. However, biology is messy, and this process is never perfectly efficient. Not every single stem cell will successfully make the transition. Some might die, and others might remain undifferentiated. Because of this, scientists need a way to measure how well their chemical signaling protocol actually worked. We need to calculate the differentiation efficiency percentage to evaluate the success rate of our biological transformation experiment. To quantify this success, we use a straightforward efficiency formula. Conceptually, this is just finding the fraction of cells that did what we wanted them to do and turning it into a percentage. Let us look at the variables. The variable E represents the overall differentiation efficiency, expressed as a percentage. The symbol NDIF stands for the number of successfully differentiated cells, the ones that actually turned into our target tissue. The symbol N total represents the initial total number of stem cells that were subjected to the chemical treatment. If NDIF increases while N total stays the same, our efficiency percentage goes up, meaning our chemical treatment was highly effective at guiding the fate of the cells. Let us apply this formula to a real laboratory scenario. Suppose a scientist treats a starting batch of 50,000 embryonic stem cells to turn them into motor neurons. After a week, they count 35,000 successful motor neurons. We need to find the efficiency. We start by placing our 35,000 successful cells in the numerator and our 50,000 total starting cells in the denominator. Dividing the successful cells by the total cells gives us a raw decimal value of 0.70. Finally, we multiply this decimal by 100 to convert it into a standard percentage. Our final answer is an efficiency of 70%. This physical result means the chemical protocol was highly effective, successfully converting a large majority of the blank slate cells into desired neurons. We have covered a lot of ground today, so let us review our key takeaways. First, embryonic stem cells are the ultimate biological blank slates. They are pluripotent, meaning they can differentiate into any cell type across the 3 primary germ layers. Second, they possess the unique dual ability to self-renew indefinitely and to differentiate on command. Third, by understanding the mathematical dynamics of their exponential growth, scientists can carefully control their expansion in the lab. You now understand both the biology and the math behind these incredible cells. Mastering this knowledge brings us one step closer to repairing the human body from the cellular level up.